Today we're going to start Chapter 7. We're going to cover chromosomes and cellular divisions. We're going to talk about DNA, we're going to talk about how it divides within the cell, and we're going to talk about mitosis and meiosis. First, we're going to start off talking about some scientists. First, we have Wilhelm von Willdyer-Hartz, and he's going to be born in 1836 in Germany. He's going to die in Berlin in 1921. He's going to attend the University of Göttingen in 1856, where he went to study math and natural science. But he switched to medicine once he met Jacob Henle, and this is going to be the same scientist that discovered the loop of Henle that is in the kidney. That's going to be found within that nephron. He transfers to the University of Greifswald in 1859, but received medical degree from the University of Berlin in 1861. He passed his medical exam in 1862. He became a university professor of anatomy and physiology at the Königsberg in 1862 to 1864. Then he transferred to other colleges that you see here. He's going to have numerous publications, over 250 of these, and he makes these publications on different anatomical topics related to cancer, hernias, and the human pelvis between the times of 1862 and 1920. So he's going to be a well-known published author on these subjects and these topics. He's going to coin several terms that are still going to be used today, most notably the term chromosome. He coined this in 1888. He also is known for coining the term neuron in 1891, so we still use both of these quite often. He was raised to nobility by the Prussian House of Lords after resignation from the University of Berlin in 1916, so he became a noble. So with the chromosome structure, we're going to have Walter Fleming, his first one to observe and describe chromatin that's going to be found within that nucleus. And he does this while using a new staining technique that was developed in 1879. Now, chromatin is going to be the form that DNA is found in during interphase or during a cell's natural life. So it goes through different phases, which we'll discuss. Most of the cells is spending its life in interphase where it's just doing its job. So that chromosome is going to be in a loose form that kind of looks like strands. I guess you could resemble it to strands of spaghetti. It only condenses into a chromosome once it's getting ready to divide and go through mitosis or meiosis. So here with the chromatin, it's going to be a mass of long thread-like fibers that are composed of DNA and proteins that's found in the nucleus. Now, these proteins are called histones, Histones. And so it's going to be wrapped around those. It's going to shorten and coalesce into a chromosome when it gets ready to divide. So changes, movements, and distribution of chromosomes when the nucleus divides are going to be observed and described by Fleming in a process called mitosis. Chromosomes are going to be composed of long continuous pieces of DNA of various types of proteins that are going to make up the organism's genome. The term is first going to be introduced in 1888 by Wilhelm von Willdyer-Hartz, and it's going to transmit its inherited traits as outlined in a chromosomal theory of inheritance. And that was introduced by Theodore Boveri and Walter Sutton in 1902. And later it's going to be confirmed by Thomas Hunt Morgan in 1910. And so they come in, they are able to now see this chromatin, and then they're able to go through and see cells that are dividing and see the chromosomes. And so they're going to determine that this is where inherited traits come from. Now, chromosomes are going to vary in shape, length, banding patterns, and the number of them is going to be different in different species. And the number of genes that's contained in them will also be different depending on the species. For example, humans are going to have 46 chromosomes. We get a set of 23 from our mother and a set of 23 from our father. With those, you have chromosomes 1 through 22. Those are called autosomes. So you have 2 of each, one from mom, one from dad. So chromosome 1, you have 2 of those. Chromosome 2, you have 2 of those. They usually start out fairly large. And as you get down through chromosomes 21, 22, and so forth, they get a lot smaller. Once you hit the 23rd chromosome, that 23rd chromosome is referred to as your sex chromosomes. And so that's where you get the XX or XY. Mother can only give an X. The father's sperm can have either an X chromosome or a Y chromosome for that 23rd chromosome. So going on and looking at this, you can get too many or too little chromosomes due to what's called non-disjunction during division. And so it can pull too many over to one side when it's dividing that cell, or it might be one of the cells that is missing some chromosomes. So this can lead to chromosomal disorders. And we have a diagnostic tool that we can look at these. So if you're taking your chromosomes and you're looking at them, you can view them in what's known as a karyotype. And that's where you can look at those different banding patterns. That's going to be a visual representation of the organism's chromosomes. And like we said, it's arranged by shape and size and staining patterns. So it's used to diagnose any type of chromosomal disorders, and they can do that with 5 main steps. The first step, the cell is going to be collected by means of blood samples. It could be amniocentesis, or it could be a chorionic villus sampling. Second step, the cells are going to be encouraged to divide. They're put into a nutrient-filled culture tube, and so that encourages them to start dividing through mitosis. Step 3, that cell division is going to be stopped when the chromosomes are going to be highly condensed and visible during metaphase. That's when they line up across the middle or the equator of the cell, right before cell division. The 4th step, the chromosomes are going to be stained, which is going to be illustrated by their distinct banding patterns that you'll be able to see. And step 5, you have chromosomes that are going to be arranged by size, shape, staining pattern, and location of the centromeres. So this is what a karyotype looks like. So you see all of your chromosomes there. One would come from your mother, one would come from your father. So you've got 2 of each of the 23 chromosomes. Now remember, chromosomes 1 through 22 are called autosomes, and then the 23rd set is called your sex chromosomes. In this case, you can see with the sex chromosomes, you have an X and a Y. The X is going to be the larger chromosome, so that one comes from mom. Mom can only give you an X chromosome because the egg is XX, so she only has X's to give for that 23rd chromosome. The sperm could be an X or a Y. Notice that the Y chromosome is much smaller than the X chromosome. So just looking at these patterns, you should be able to immediately tell if it's a male or female. If you had 2 larger chromosomes, then you would have the XX, so that would be female, but this is large and a smaller one. Now, prokaryotic chromosome structure is a little bit different. Prokaryotic DNA is going to carry genetic information, just like with eukaryotic. It's going to have what's referred to as a nucleoid-associated protein, and that's going to function in gene regulation and chromosome organization. It exists as a large, single, circular piece of DNA that's going to be located within the nucleoid region of the cell. So it does not have a nucleus that surrounds it. It just has a specific region where it's more likely to be found, and that's called the nucleoid region. Due to its size in comparison to the overall dimension of the cell, the chromosome is going to be tightly packed, and that results in a genome with densely packed genes in it. So the eukaryotic chromosome structure, remember these are going to be plants and animals, protists, and fungi. With the prokaryotes, those were your bacteria and archaea. So now we're going to what would be inside of you for an animal. It's going to be located within the nucleus of a cell and composed of DNA and proteins and masses of threads and fibers called chromatin. Now, what that chromatin is found free-floating within the nucleus and consists of a single molecule of DNA and histone proteins that is going to be wrapped around. Now, with the histone proteins, they're primarily going to play a structure role. They prevent the DNA from tangling up, and they help to organize those chromosomes and helps them to tightly pack when they get ready to start dividing through mitosis. So let's go here. I'll get my pencil. So you've got your DNA, and you can see with the DNA strands, they're going to be wrapped around these clusters those clusters are going to be your histone proteins. And so as it wraps around them, it's going to keep it from tangling up or mixing up any of those nucleic acid base pairs that you see there. Okay. So DNA packing results in what's known as nucleosomes. That's going to be a bead-like structure that's containing DNA that's double wrapped around 8 histone proteins. So that's what you see in that circle where you see histones are pointing at it. Additional folding and coiling of the chromatin is going to result in what we call a chromosome. And so that's that condensed shape or that X shape that we look at, and we just look at and automatically think, hey, there's a chromosome. It's formed from chromosomes containing a single centromere and 2 telomeres. And so if you look in the center portion, that's where your centromere is going to be within that chromosome there. At the end of the DNA strands, or the end of the chromosome, it's going to be capped with telomeres. And those telomeres are going to be protective sections of bases that are there to help to protect the DNA. And as it starts to divide, it kind of also will be a counter, kind of like an odometer. And it's going to count how many times that cell divides. And so as it's dividing, it only allows that DNA to divide so many times before it stops the division of that DNA. Because anywhere from 50 to 80 divisions is going to be the sweet point or the good point. If it divides much more than that, then it will actually start to cut into those telomeres, and you will start to see more mutations and more mistakes being made with the duplication or replication of those DNA strands. All right, so with that centromere, that's going to help to attach those sister chromatids. And so what makes the chromosome is going to be 2 chromatids, and they're called sister chromatids. And so it's going to go in, it attaches, and it helps it to kind of be held together like that. And sister chromatids are simply going to be replicated DNA, so they are matching chromosomes. So they match one another after DNA replication, and that occurs during S phase of interphase. And it's going to be there to help it start to separate during cell division. So that's the reason that we have that centromere there. It has to double up its DNA before it goes through mitosis and makes 2 identical daughter cells. So with the telomeres, like we said, it's going to consist of repeating 6 nucleotide sequences that are located at the ends of chromosomes, so they're protective ends. They are not coding for genes, so they lack for genes, and they do not code for any proteins. They're going to help to maintain chromosomal stability by preventing the removal of coding proteins of that chromosome. So it's like little safety end caps on the end of each sister chromatid. They also act, like I said, as an internal clock or a odometer. They're going to specify the maximum number of divisions that that cell can undergo before cellular death. Now, the correct scientific name for cellular death is going to be apoptosis. So know that term. That idea became known as the Hayflick limit. That was named after Leonard Hayflick, who discovered this phenomenon in 1961. So according to Hayflick, that limit, a human cell can divide about 50 times before it starts to lose functional DNA, and then that results in apoptosis. Like I said, with some cells, it can go up to 80. Cancer cells are going to continually divide because they can rebuild and extend their telomeres using enzyme telomerase. And so sometimes the longer those enzymes are, the faster that DNA divides, and it can create cancerous cells. So with this Hayflick limit, you can see how that one cell divides into 2 daughter cells. Those 2 divide into 4, and so forth. And you see the little capped ends, which are the red ends. Those are your telomeres. So the more it divides, if you look up top, you've got a nice full telomere here at the end. As it divides, that telomere starts to break down more and more and more. Once it gets to its maximum number of divisions that it needs to go through, then it will stop, and it goes through apoptosis at that point. For prokaryotic cells, They don't go through mitosis, but they go through a process that's going to be very similar. This process is called binary fission. So it's a form of asexual reproduction in which one cell divides into genetically identical cells. So it makes one parent cell into two daughter cells. It can only occur with adequate supply of nutrients and optimal temperatures. Those are going to be the key. The majority of prokaryotic cells can complete cell division in 1 to 3 hours, while others can perform this process in as little as 20 minutes. So the well-known E. coli bacteria divides about every 20 minutes. Two main stages of binary fission. First, you have your cell growth and chromosomal replication stage. So what happens here is those prokaryotic cells are going to grow, they increase in size, they double up on everything that's in there. So they'll double up on their cytoplasm, they double up on their chromosome that you see there. They have special locations on the cell membrane that start to form, and then they start to divide from that point. And so you'll start to see them trying to get my pencil. You'll start to see them pinching in at these points. Those are called cleavage furrows. And then it pinches in to the point that it's able to separate itself from one another and then pops apart and divides. Once it pops apart and divides, that is referred to as cytokinesis. So with binary fission, it occurs when the DNA replicates and consists of three main steps. After the DNA replicates, So what first happens is your cell elongates. You saw that in that previous picture. As the length of that cell increases, the attachment points of the chromosomes begin to increase. And so that pulls those chromosomes apart. New cell membranes and cell walls are going to begin to form. So that occurs as the cell continues to elongate while proteins begin pinching that cell in at that midline. Eventually, it's going to form two daughter cells. So that completes the binary fission as the parental cell membranes and the cell wall continue to pinch in and then fuse together. And then with each new daughter cells produced and replicated chromosomes, it makes them genetically identical to that parent cell. That's the cell that first started out. And so looking at it again, you can see how it is starting to double up on everything. It goes through that elongation phase. Then you have those new cell membranes starting to form in here where it's pinching in. That cell membrane and cell wall is completely formed, and then it can pop apart and form two daughter cells in a process known as cytokinesis. Advantages of binary fission are that they're fast and they're easy to perform. Each daughter cell is going to be genetically identical to the parent cell. It's going to allow prokaryotic cells to quickly establish new populations if their numbers are reduced, and enables them to reproduce in isolated habitats. Disadvantage is it produces genetically identical offspring, so if those offspring are not good for whatever environment they're in, then they could die out. So that reduces their genetic diversity. It makes them less adaptable to different environments or environmental changes that could occur. Now we're going to go into the cell cycle. And that's going to be a series of three main events in which the cell performs normal cellular processes. And in the end, you're going to end up with the cell dividing into two daughter cells. So looking here, we have the interphase area. Interphase is going to consist of cell growth, DNA synthesis, and then more cell growth. So you have what's referred to as G1, DNA synthesis or S phase, and then G2 phase. Throughout this, you will have several checkpoints, and then it goes into mitotic phase where it divides. So the first step in the cell cycle is once thought to be the resting phase of interphase. That's going to be an active period actually for the cell. That's where it performs its normal cellular functions. That includes things like metabolism, making organelles, synthesizing proteins, duplicating chromosomes, and just simply growing and doing what it does as a cell. It's going to spend about 90% of its time in interphase, which is about 20 hours. The four main phases, I didn't discuss G0 at first, But G0 is going to be a distinct dormant stage for some of these cells, like muscle cells and nerve cells. They're the ones that don't really go through mitosis. Upon reaching maturity, these cells will remain physiologically active, but they no longer have to go through cellular divisions. So these, once you have your muscle cells formed, once you have your neurons formed, that's pretty much all you're going to get. They're not going to go through division and create more. G1 phase is going to be a period in which a cell grows, it synthesizes proteins, and it carries out cellular respiration. In addition to synthesizing and gathering materials that are required for DNA replication and cell division. During this, you can go through what's known as a G1 checkpoint prior to entering S phase of interphase. That phase is going to basically, or that checkpoint's basically going to check to make sure everything's running along smoothly. If it is, it will enter the third step, which is going to be S phase, and that's a period that's associated with DNA and centrosome replication. So think of S phase and synthesis phase. Here, DNA is replicated, and it duplicates those chromosomes, results in sister chromatids, which are matching chromosomes, that are joined together in the center with the centromere. Centrosomes are going to be organelles that help to organize the microtubules and structures that are going to be made to form that tubulin, which is going to be responsible for constructing the mitotic spindle. Centrosomes were independently discovered in the 1880s by Edward van Bendenen and Theodore Boveri. Only are going to be found in animals. So when we looked in lab at animal cells and plant cells, you saw that you had a centrosome and centrioles that are going to be within that centrosome. Those are what are going to help these cells divide and help these chromosomes migrate to the poles, which are going to form the new cells. So centrioles are going to be the organelles that contain tubulin and function in organizing the mitotic spindle and help with the cytokinesis. G2 phase is going to be the final phase of interphase, and that cell is going to be actively synthesizing proteins and cell membrane materials in preparation for cell division. So these cells are going to pass through the G2 checkpoint prior to entering mitosis. That G2 checkpoint is going to make sure that the chromosomes are going to be replicated correctly and everything's ready to start dividing. Now you're leaving interphase and you're entering the M phase or mitotic phase. So here is mitosis. That's the second step in cell cycle. So first step was interphase. We just talked about the 4 parts of interphase. Now we're hitting the second step, which is your mitotic phase or mitosis. The entire process was first observed and formally described by Walther Fleming in 1879. Anton Schneider is going to be the first observed and described metaphase and anaphase in 1873. In 1882, Walther Fleming is going to give a more extensive description of mitosis, which he describes as occurring in 2 phases, the progressive phase and the regressive phase. So in his progressive phase, that's going to be where the chromosomes are going to be visible and aligned in the center of the cell. Now, this day and time, we call that metaphase of mitosis. The regressive phase is going to be where the chromosomes are going to be separate and the cell divides. So with the mitosis or M phase, it's going to be divided up into 4 main stages. And this takes no more than around an hour for it to completely go through these phases. So the first step is going to be prophase. That's where the chromatin, remember it's going to be that stringy DNA, is going to condense into chromosomes, so now you can see them. The mitotic spindle fibers are going to start to form. The nuclear envelope fragments are going to start to break down, and then you're going to have the chromosomes attached to the mitotic spindle. So once that nucleus disintegrates, basically, and that spindle goes to each end, it's going to come out and attach at that centromere of the chromosomes. So fragmentation of the nuclear envelope, like we said, releases that centrosome into the cytoplasm, which quickly is going to be bound to what's called the Ki67 protein. Now, these proteins are going to prevent the chromosome from coalescing once released from the nuclear envelope. The second step of mitosis is metaphase. This is condensed chromosomes that are going to line across the center of the cell along the region known as the metaphase plate. Lots of people refer to it as the equator of the cell since it goes across the center of the cell. So you see them line up. You see that you have the centrioles on each end. They have their spindle fibers that are coming out. They're going to attach in. Remember, with your chromosomes, easy to just put an X there and put that centromere in the center. It's going to come in and it's going to attach to the center portion of that centromere. And it's going to be able to kind of break these chromosomes apart or pull them apart. And so one half of the chromosome is going to go to one end of the cell. The other half of the chromosome is going to go to the other end of the cell. And what's going to happen is these spindle fibers are going to start to shorten in order to pull them apart. There we go. Needed to switch slides. All right, third phase of mitosis is anaphase. That's where your sister... Oh, now it's catching up. Must have just had a lag. Okay, the sister chromosomes or sister chromatids are going to be pulled apart by the centromeres or the spindle fibers attaching to the centromere, shortening, and so that pulls each chromosome to either one end of the cell or the other. So you see that mitotic spindle starting to shorten. Also, the cell is elongating, so it's getting longer. The fourth phase is going to be telophase, and so that's your last phase of mitosis. That's where the chromosomes are going to start to uncoil. The mitotic spindle is breaking down. You have a new nuclear envelope that's starting to assemble around these chromosomes and form two new daughter cells with their own nucleus inside. Nuclei is going to be plural for nucleus. And so here you see those two forming. Where it is pinching in through here, that's referred to as your cleavage furrow. It's going to pinch in to the point that it pops apart. And once it does that, that's a phase called cytokinesis. So mitosis is technically the division of the nucleus. Cytokinesis is the division of cytoplasm. So that's where it actually pops apart and makes two separate daughter cells. Remember, the original cell we started with was called the parent cell. So that third and final step of the cell cycle, the division of the cytoplasm, cellular contents, and that newly formed cell is going to occur, and that's what's called cytokinesis. Generally, the cell then continues until the G1 phase of the next interphase starts. This process can differ in animals and plants just depending on which one you are talking about. So cytokinesis actually begins in anaphase, and it's also known as cleavage. So it starts to, once it elongates, bend in on the sides, and that's where you see those cleavage furrows. So proteins actin and myosin are going to form what's known as a contractile ring, and that is going to constrict and create that indentation known as the cleavage furrow at the end of metaphase as it's going into anaphase. And so it's going to be right there where that metaphase plate was. As the contractile ring continues to constrict, because of those proteins, those are contraction or contractile proteins, the parent cell is going to be pinched into two daughter cells with their own nucleus and their own cellular components. That means their own organelles, their own cytoplasm, everything they need to exist as their own independent cell. If it is a plant cell, it's going to start as a cell plate. It doesn't actually go in and create that cleavage furrow. And it also will not have spindle fibers, and it's not going to have those centrioles or centrosomes. Remember, those are only found in animal cells like what we just saw. So we're going to form a cell plate, which is going to form around that metaphase plate. The vesicles that contain carbohydrates and components for a new cell wall and cell membranes are going to be released from the Golgi apparatus. Vesicles are then going to congregate and fuse together, and that's going to form your cell plate. As more vesicles start to congregate along that cell plate, new cell walls are going to begin to form and divide that parent cell into two daughter cells. Important reproductive method in some unicellular organisms such as yeast, amoebas, and parameciums. In multicellular organisms, this process is going to be important in growth, maintenance, and repair of body tissues. And so for an organism like you, you're going to use this to grow and maintain everything that you have and repair any damage. If it is a unicellular organism, this is simply going to be how it creates new organisms. In biotechnology, it's going to be used for the purpose of cloning and generating stem cells. So all of that we just described was how cells divide or reproduce asexually. Now we're moving into sexual reproduction. This is going to be a mode of reproduction that involves the fusion of gametes from two parents during fertilization. Fusion of gametes during fertilization was first described by Oscar Hertwig in 1876. Gametes contain only half of the number of chromosomes as somatic cells. Somatic cells are going to be your body cells. Gametes are going to be sperm or egg. And so a discovery that was made by Eduardo van Beneden in 1883 described gametes being haploid, where you have a lowercase n, and somatic cells being diploid, which is a 2n. So remember, haploid is half of the genetic information. So remember, you have 23 pairs of chromosomes. So the haploid number would be 23. The diploid number, since it's pairs, would be 46. So human somatic cells have the 46 chromosomes, which is diploid, while their gametes are only going to have 23 chromosomes, which are haploid. So a regular cell, a kidney cell, liver cell, skin cell, 46 chromosomes. But the egg and sperm only have 23. Of the 23 chromosomes in a human gamete, 22 of those are going to be called autosomes. And the remaining chromosomes are your sex chromosomes. And we kind of discussed this a little bit earlier when we were talking karyotype. With the sex chromosomes, you're either going to have an X or a Y. They're going to differ in size and genetic makeup to determine the sex of the offspring. So here's your karyotype again that you're looking at. That's what this picture is. And at the bottom, you can see your 23rd pair, the sex chromosomes, are going to be a large chromosome and a smaller one. So that's going to be an X and a Y because the X is going to be larger. When haploid gametes fuse during fertilization, a diploid zygote is going to be formed. So you're taking 23 chromosomes from the egg, 23 chromosomes from the sperm, you put them together, and you end up with 46 chromosomes. Remember, 46 is your diploid number. As soon as it's fertilized, it's referred to as a zygote. That's a phenomenon that was observed by Eduard van Beneden in 1883. So the zygote contains genetic information from both parents and develops into a genetically unique adult. Gametes are going to be produced in the ovaries and the testes of sexually reproducing organisms by a process known as meiosis. So Eduard van Beneden is going to observe and describe meiosis in 1883 while studying horse interstitial roundworms. And so with that, he looked at the specific worm here that you see in the picture called Ascaris. Ascaris is going to have a total of 4 chromosomes, but van Beneden discovered that the gametes contained only 2 chromosomes. Once the gametes fused during fertilization, the original chromosome number was restored. So it went from the haploid number to the diploid number. So it went from 2 to 4 again. So meiosis is going to be preceded by interphase, a 3-phase process like mitotic interphase. It's going to have a G1 phase, which introduces the cell growth, protein synthesis, organelle replication, and synthesizing materials required for DNA replication. S phase is going to be where replication takes place of that DNA. So each pairs of chromosomes, known as homologues or homologous chromosomes, are going to replicate to produce sister chromatids that are going to be attached at the centromere. Homologous chromosomes are inherited from each parent. In human offspring, we inherit, like we said, 23 from mom, 23 from dad. Similar in length, shape, and staining pattern, and centromere position. They also have a similar genetic composition and gene arrangements. That means we can find, let's say, genes for hair color, height, eye color, things like that, on the same chromosomes around the same place. The centrosome, the original or the organelles responsible for organizing the spindle apparatus, microtubules, are then also going to be replicated. So you see one on each side there. So those are your centrosomes. Let me get the... Now, what I forgot to mention during mitosis is that when these chromosomes duplicate themselves, they're going to duplicate from 46 to 92. So remember, they're doubling up. So you go from 46 chromosomes in an original haploid cell to 92 chromosomes. So when they divide, then each daughter cell ends up with 46 chromosomes again. But with meiosis, it needs to divide into 4 cells. And so remember, it's got to reduce the number of chromosomes down by half. So instead of having 46, we're going to need to have 23. So when it goes through its first division, or gets ready for its first division, it still needs these 92 chromosomes, just like it did in mitosis. So with meiosis, it's going to proceed by interphase, a 3-phase process, just like with the mitotic interphase. So we had the G1, we had the synthesis, now we're going into G2. That's the final phase where the cell is preparing for cell division by synthesizing its proteins that are associated with that spindle apparatus and producing materials that are required for building new cell membranes. So meiosis is going to occur in the ovaries and testes only of sexually reproducing organisms, and this is in order to produce gametes. So it consists of 2 nuclear divisions. Each is going to contain 4 phases, and it's going to be responsible for reducing that diploid cell number into 4 genetically distinct haploid cells. So these are no longer going to be genetically identical. So your first phase, or your first set of phases, is going to be called meiosis 1, and then your second set is going to be called meiosis 2. So in meiosis 1, the first nuclear division is going to separate those homologous or homologous chromosomes into 46. By the end of meiosis 2, you're going to have a second nuclear division, which is going to separate the sister chromatids in a process called, or process like mitosis. So it goes there from 46 in each cell to now 4 cells with 23. So with meiosis, we're going to divide twice with these cells. So you're going to go through what's known as meiosis 1 and meiosis 2. So we're starting out with the first phases of meiosis 1, and that's going to be prophase 1. Here, we're going to have homologous chromosomes that are going to start to condense. So they're starting to tighten up, just like what we saw in mitosis. They're going to become visible, and then they're going to align and attach with each other along similar genes to form these things called tetrads. These tetrads are going to contain what we know as replicated maternal and paternal chromosomes that are going to equal 4 chromatids. So that's where it gets its name tetra from, 4. During this phase, crossing over can happen. This is going to be very important. You will be asked questions about this. So what crossing over is where these chromosomes line up, both the mother chromosomes and the father chromosomes, and they can actually recombine their DNA or genetic material randomly. So, get my pencil. You have one chromosome here, and then you can have a chromosome here. Well, they could get really close, and the leg of one can switch with the leg of another. And so then, let's say chromosome from mom might be all mom material except for this little section down here, and dad's chromosome might be all dad's DNA material except for this little section of the chromosome here. And so with that, that's where they can switch up and become even more genetically diverse. So it's just a random exchange, and that only occurs during prophase 1, and it is called crossing over. So make sure you know what crossing over is. So they line up along that region, they touch, and that area where it occurs is going to be known as the chiasma. And so that's where crossing over happens. So crossing over is going to assist with the separation of chromosomes and helps generate genetic diversity, like we said, because we're mixing up mom and dad's genetic information. From there, that nuclear envelope fragment, the spindle apparatus is going to form, and the homologous chromosomes are going to attach to the spindle by these proteins called kinetochores. And so here we're looking at it. We have the mom and the dad's genetic material. Let's have dad be in blue and mom be in red. They get close together. Those bottom legs that we see there of the chromosomes cross over, touch. They're going to switch genetic information, and then that's how it can mix up that diversity even more. Like we said, only happens during meiosis 1 and prophase 1 is the specific phase. Next, you're going to go into the second phase, which is metaphase 1. That's where those condensed homologous chromosomes are going to randomly and independently align in the center of the cell along that metaphase plate. So you see them line up. Now, when they do this randomly, the mother's doubled up chromosome and the father's doubled up chromosomes are going to line up. You might have some of dad's on one side and some of mom's on the other side. So it's not all dads on one and all dads on the other. So they just randomly line up. This also increases the chance of genetic diversity. So some of these genes are going to go towards one side where other genes may go towards another side. That's why every egg that's created and every sperm that's created is going to be genetically diverse. So you can have two parents that have, let's say, 10 kids, and they all will have some similar traits, but they all will be genetically different because of all of this genetic diversity. Anaphase 1 is where these homologous chromosomes are going to randomly start to separate as the spindle fiber that came out and attached at that kinetochore is going to start to pull it to either one end or the other. And so now we really start to see the mixture of maternal and paternal chromosomes go into these opposite poles. So unlike mitotic anaphase, these sister chromatids are going to remain attached to each other at this time. So all of them were attached using a kinetochore. Now we've separated them. We're going to go into a cell that has 46 chromosomes each that are attached in that center section. Telophase 1 is going to be where these homologous chromosomes are going to start to uncoil. The spindle apparatus is going to break down, and the nuclear envelope may or may not reform around each haploid set of chromosomes. So it may come together or it may directly go into its cytokinesis and next phase. So with cytokinesis, like I said, that's going to be the division of cytoplasm. So we have that cleavage furrow starting to come in and it forms, and of course this is only in animal cells. You have a cell plate that's going to form that goes through the plant cell. And so with those daughter cells with haploid numbers of chromosomes are going to be there. So cells may return to a short interphase after cytokinesis. During this interphase, those chromosomes are going to temporarily uncoil, but they're not going to replicate again this time. They are not going to go through that S phase. And so the proteins are going to be synthesized that are going to help these cells proceed to meiosis 2. Going into meiosis 2, we're going through the same steps, except for you're just going to have a 2 behind them now. So we're going to start out with the prophase 2. That's where the sister chromatids that were condensed are going to recondense, and you're going to have new spindle apparatus starting to form, and that's coming out of those centrioles. The nuclear envelope is going to fragment and start to break down and dissolve, and the sister chromatids are going to attach to the spindle using those kinetochores. Sister chromatids, which are generally going to be distinct, are then going to be pulled by the spindle apparatus after they align along the metaphase plate. So remember, they line up across that equator around the center. The spindle come in, they attach to the kinetochores, and now we're going to separate these sister chromatids. Anaphase 2, the sister chromatids get separated because these spindle fibers are going to start to shorten, and it's going to bring these towards the poles. So they go towards opposite sides of the cell. Telophase 2, you start to see it cleaving in, and so the chromosomes start to somewhat uncoil during this period. That spindle apparatus breaks down, and the nuclear envelope is going to start to reform around the chromosome. Eventually, it's going to have such a cleavage furrow to where it pinches in, it's going to completely divide. And by the end of this, you're going to have 4 distinct haploid daughter cells. Now, they are going to be different from the parent cell, and none of these cells are going to be genetically identical like they are during mitosis. They're all genetically diverse. So going into gametes. First, we're going to talk about the sperm. It's going to be small, a lot smaller than the egg. The egg's going to be much larger, contains more cytoplasm, organelles, things like that. It's also going to be motile. That means it can move. It's going to move using a flagellum. Equal size cells containing little cytoplasm there. So it's going to have the DNA that's going to be found in the center inside of a nucleus. That's going to be found in what's known as the head portion of the sperm. Then you're going to have a neck piece that comes along this way. It's going to have ATP and energy, and that's going to help to propel or move this flagellum here. And so inside with that DNA that you find here, it is haploids. There's only going to be 23 chromosomes there. So the sperm's first observed in 1678 by Antony van Leeuwenhoek. And he's going to believe that they're actually parasites. He knows preformation as an idea, and so this gets developed during this time by Leeuwenhoek in 1685, and that's going to suggest that the sperm contains a pre-made human that just relied on females for nourishment. So they think that inside of the sperm here, you already had the full pre-made human being. And inside, it was delivered to the female body, where it then grew and became nourished until it was ready to be born. And so that is going to be the structure that he suggests there. Experiments performed by Lazzaro Spallanzani in 1779 suggest that the sperm was responsible for fertilization, but he determines that it's not going to be microbial contamination, but an important part of fertilization in the 1800s. So it's not an actual contamination or a parasite, as Leeuwenhoek first thought. Now with the egg, it's going to be much larger in comparison to the sperm. It contains a lot more cytoplasm. 4 cells are going to be produced in the ovaries during meiosis, but only one of those cells is actually going to reach to maturation. So most of that cytoplasm, as it's dividing from 1 cell to 2 and then 2 to 4, most of that cytoplasm gets pushed into one main cell, and that cell's going to end up becoming the egg. The other 3 that don't get used, that have the less cytoplasm, those are going to become known as polar bodies. These will degenerate and become reabsorbed by the female body after they form. And so they start to break down, and then anything that could be reused by the body can take that broken down substance and reuse it. Going into cancer research, we're going to have a very important woman. Her name is Henrietta Lacks. She is going to be what we know as the immortal contributor to biochemical research in the 1950s. And so 1951 to be exact, she goes in during treatments. She's suffering from cervical cancers. They're going to take a sample of her cervical cells that have this cancer, and they're going to find out that these cells are immortal. They don't die. These are going to become known as the HeLa cells. These cells are going to be the first group of immortal cells to be cultured and grown in a laboratory setting. Continuing to thrive and multiply today, we still have these cells in a lab. The HeLa cells have been widely used to study genetics. They've been used to develop vaccines such as polio and the COVID-19 vaccine, and it determines the treatment for things like AIDS, Parkinson's disease, and infertility. And they're also going to be useful and important in the study of cancer. So it led to many numerous discoveries, such as the link between HPV and cervical cancer, or the human papillomavirus. Cancer is going to be defined as an uncontrollable cellular growth and division. It's going to result in cells that can spread into surrounding tissues. It damages them by taking up the nutrients, taking up their space, and basically choking those cells out until cell death. It occurs when a cell loses the ability to control its cell cycle because of a mutation that leads to a cell ignoring its apoptosis signals. The telomerase is going to be the enzyme that's produced by cancerous cells that keeps rebuilding their telomeres, that tells them, hey, you need to self-destruct, you need to go through apoptosis. That's what's responsible for regulating cell division. So characteristics of cancer, it's non-specialized cells usually. They have an abnormal shape, indefinite division. They quit doing their job and keep going through mitosis over and over and over again until they make a mass. That mass is referred to as a tumor. They have large abnormal nuclei. They are immortal, so their telomerase is going to help cells bypass that apoptosis by not letting it break down the telomere and not letting it tell itself to self-destruct. They're going to lack contact inhibition. That means when they become too close to another cell, most cells will stop growing, they stop dividing. These cells do not. They will start to clump up and build up on top of each other, and that's going to form your mass, which is the tumor. They can metastasize. So metastasis is going to be the lack of adhesion molecules, so that means that they can move around the body. That's going to be dangerous because if it can move to other areas, it's going to spread these cancerous cells and cause tumors and cancers in those other parts of the body. And they have angiogenesis. That's going to be formation of new blood vessels within healthy tissues or organs, and that's to feed these masses. And so now they're getting unlimited food supply, they're getting plenty of oxygen, and they're carrying away their waste products and carbon dioxide. And they can be transmissible between individuals of the same species and different species. Tumors are going to be defined as abnormal masses of cells that no longer are going to be regulated by the cell cycle. Now, you can have two types. You can have what we call benign tumors. These are sometimes called non-cancerous or non-metastasizing. Examples of these would be like a uterine fibroid, lipomas in the skin, things like that. Most cancers are going to begin as benign tumors. Usually, let me go back here, this benign tumor, the body comes in and it will wall off this area and encapsulate it. And so that keeps it from moving around. So the body can pick up, hey, this isn't right, these cells aren't right. It didn't get rid of them, but it can wall them off and surround them in a capsule. Malignant tumors are going to be masses of cancerous cells that can metastasize and invade healthy tissues or organs, so they can move around the body. Once cancerous cells have invaded healthy tissues or organs, they initiate that angiogenesis, which means that they're going to have more blood vessels coming to that area to help feed them and help them grow. And so that affects the normal physiology of the tissues and the organs. Now it's sending all of its nutrients and all of its resources to the cancer. Treatment options, you can have surgery. That's your first step. You need to remove the cancerous tumor, and typically it's the most successful. Not effective against metastatic cancer though, because that means it's already gone into either the bloodstream or it could go into the lymphatic system and it's started to move around the body. High energy radiation is going to treat localized cancerous tumors, but not against metastatic cancers, the ones that have moved around. It's effective in damaging the DNA of those cancerous cells and affects their ability to divide and grow. Also, it's going to damage the DNA of normal, healthy cells too around it, but these cells can repair their damage, and characteristics of the cancer cells don't have. They're not able to repair the damage. You can treat it with chemotherapy. This is going to be the most frequently used cancer treatment. It's effective against metastatic cancers. It involves a use drugs that are called analogs. And what they do, they're structural derivatives of DNA and nucleotides. So with these, the cancer cells are going to be used as analogs for DNA replication, but it's going to go in and disrupt the process, leading to the inability of these cells to divide. Therefore, once they stop being able to divide, it's going to slow the tumor growth. It can affect healthy immune system cells, though, so it makes a person receiving that treatment more vulnerable to infection. Patients that are exposed to chemotherapy have reported easy bruising. They've reported a lot of nausea and loss of hair. So that's going to end us for chapter 7. and we'll pick up with Chapter 8 in our next video.